of the variability of the trait is related to genetics, but probably this 25% increases at increasing age. So in centenarians and in supercentenarians, genetic probably play also a major role, more than 25%. Centenarians, nonagenarians, say first degree relative have a lifelong 30% survival advantage. Members of long-lived families have better metabolic profile and have a better inflammatory profile. And this was the basic study. So we recruited SIB pairs, so brother and sisters belonging to the same family, which were both, or even 3 or 4, older than 90. And this was a control from the same region, geographic and ethnic origin. We did linkage studies which looked for the coinheritance in families of alleles shared by which are identical by descent, and test whether siblings affected in this case by longevity share more alleles transmitted to them from the same parent than expected by chance, so higher than 50%. This type of studies is more powerful than association studies to identify rare, high-risk alleles, so rare but very powerful alleles. And of course, this is the idea that in this case, the two SIBs do not share any alleles, in this case 1, in this case 1, in this case 2. So this is the background. We used this platform, this Illumina Infinium platform, which has about 6,000 SNPs, and we genotyped 2,118 couples, which means more than 4,000 people. And the 90 plus people comes from all this center all over Europe. Of course, population structure is extremely important, and you can see here that the Finnish are totally apart. The Sardinian and the Mediterranean people, like in Italy and Greece, are here, and here is the northern country, Germany, Denmark, the Netherlands, and so on. So in order to do this type of study, you have to take into account the population structure, which means the population history and so on. And this is the result of the genome-wide linkage study on all these people. And you can see that there are some peaks over a log score of 3. And these are the peaks that we found. In blue are the non-parametric linkage, and in yellow are weighted for linkage. So the families which are have members, the SIBs are older than the mean, and we found three major regions and 4 peaks: the first, the second, and the third, in different chromosomes. And what is interesting is that only in one case, in chromosome 17, the age-weighted linkage is higher than the mean. And these are the chromosomal region, the 14q11.2, the 17q12, and this in chromosome 19. One of these peaks is related to ApoE, and this is very important because this is the first time that a genome-wide study, is able to identify ApoE, which was very well known before, but this means that the linkage is very powerful. And this gives you an idea of the contribution of the different European regions, and you can see that in most of the cases the northern countries contributed more than the southern one, apart from here that both contributed. Then, in order to, since these regions are quite wide, and under this region there are about 1,000 genes, we thought that to do GWAS, so genetic association studies, and we did this in more than 1,000 of the oldest member of the SIB ship with the controls. And we found ApoE as it was, as I mentioned, but nothing else. So the GWAS didn't help us in identifying something under the regions that I showed for the linkage studies. And if you put together the data of the linkage plus the association, you find not only ApoE, but ApoE plus TOM40, which is a mitochondrial gene, and ApoC1. So, in conclusion, we identified these 4 chromosomal regions, and with this new technology, we are planning to resequence entirely these regions in order to identify the genes, but we have applied to European grants. What is important is this one. Since common variants from the association studies, genome-wide association studies, do not seem to contribute to linkage peaks for familial longevity, rare and private variants may better explain the linkage in the remaining region. In other words, we think that the genes which play a role in longevity are rare variants that you can pick up looking at families at linkage studies. Something unexpected was the gradient between northern and southern Europe, for example, for ApoE4. ApoE4 is a universal risk alleles for Alzheimer and cardiovascular disease. And you can see that in Finland, in the northern countries, is about 20% of the population is positive for these risk alleles, and in Italy only from 7 to 4%. In a study that I recently published on Alzheimer, we studied more than 1,400 individuals, and we found that the frequency of ApoE variants was 8.4% in the age-matched control and 23% in Alzheimer's disease. But 23% is exactly what you found in normal population in Finland. So this means that the ApoE4 play a positive role in Finland, because otherwise would have not been selected for in that population. So the population genetics has to take into account the demography and the geography. What about in this GEA project? We studied also the mitochondrial variants, because we published in 1999 papers suggesting that DNA inherited variants are associated with